Starting from over 100.
So, my that will be actually happy because I
Hello? Hello?
Hello.
Hello?
Yeah, we can hear you.
Okay. Latif, can I take over? I believe, if you can hear me, I believe Latif may be of the out of hearing range. Hematology was supposed to be, or is supposed to be now. So I believe, yeah, okay. Ebenezer, thank you for saying you can hear me. Yes, Abdul.
Yes, Prof. You're welcome.
Okay. Okay, so what's the information I got? I try to center what I'm going to discuss around the issues that the people that were surveyed put on the platform, including the that they mentioned that they have problem with. So people talk about something on blood cells, deficiency, anemia, hematological malignancies, hemolytic anemia, and red blood cell morphology. And so I probably will start with the blood cells, and then we look at their morphology for today, see how we go. So, okay, where is this one? Right, so I guess I can share my screen now. All right, excellent. So I can share this one. Oh. This. There you go. All right, so I'll quickly talk about blood cells. And please, if I'm going at a speed that is not good for you, just draw my attention either by putting in something in the chat or raise a hand, and then I will slow down. If you also have a question midstream, you can still put it in the chat or raise your hand, and then we talk. I will try and put the talking in the not to cover all the time, leave quite enough time for question and answers, because most of what you need is actually with you, and by clearing up the questions that you have, that will be of more beneficial to you, probably. So I won't be talking throughout the time as we do with our normal lectures. So quickly on the origins and interrelationship of blood cells, as I want to talk about now, we will see that blood cells are formed in the bone marrow, all of them. When you go to immunology, they will tell you T cells develop in the thymus. But in terms of hematology, we are keen on the fact that blood cells are formed in the bone marrow, and they all come from the bone marrow stem cells, which are immortal, meaning that they will survive until the person who owns them dies. Immortal in the sense that they are self-renewing. So you have one stem cell, it develops into a second one, and then the same stem cell will remain. And that is why they are immortal. They are also undifferentiated. That is very primitive because they haven't developed into any type of the functional cells. The functional cells are the final ones that carry out the function. So in white cell, we have 5 of them: neutrophil, eosinophil, basophil, monocyte, and then lymphocytes. And then with the red cells, and then we have the platelets forming the complete cell number. So looking at this picture, if you take a section of the bone marrow vasculature, you see the stem cell, and the stem cell changes. So this is a very, very undifferentiated one. It differentiates by receiving growth signals. It differentiates into a second stage multipotential stem cell, meaning it can be myeloid, go back, or lymphoid. So at this stage, depending on the body's needs, this cell will receive signal that will convert it into a lymphoid progenitor cell, meaning it can become a natural killer cell, T lymphocyte, or B lymphocyte. Hematology, we say lymphocytes. So we combine them, because when we look at blood films, you cannot differentiate B from T lymphocyte. So we combine them. And then the myeloid lineage, if you convert to the myeloid lineage, we get a neutrophil, basophil, eosinophil, monocyte that changes to macrophage, and then platelets. Platelets are broken off from megakaryocyte. So you have a karyocyte that breaks off into the smaller platelets. You have the red blood cells, all coming from the myeloid progenitor cell. So these ones here, if you could see the cursor, that is, they are the functional cells, the ones that carry out the functions. The progenitor cell cannot do the function of eosinophil, neither can it do the function of neutrophil. All right. Then the cell needs the hematopoietic supportive stroma to be able to develop. All right. So in the bone marrow, we call it the microenvironment. That is where the cells uses the nutrients and develop into these cells. All these occur in the bone marrow. Of course, at some point, the T lymphocytes will leave the bone marrow and go to the thymus to finish their development over there. So the stem cells are immortal, undifferentiated, multipotential. Multipotential meaning they can become any of the blood cells. And they go through this development called haematopoiesis. So haematopoiesis is the development of blood cells from the bone marrow or in the bone marrow. And through the process of haematopoiesis, the cells become what is called hematopoietic stem cell or blood forming cell. And they say the hematopoietic stem cells are more differentiated than a fertilized egg, but they are not too well differentiated. They give rise to 3 types of cells that we have: the leukocytes, that is white cells, erythrocytes, red blood cells, and thrombocytes, platelets. So the hematopoietic stem cell will give you all those three. Then you come and break down the white cells into the 5 different white cells, which becomes 6 when you add macrophage, and then the thrombocytes being the platelets and the erythrocytes. So 3 main cell lines, but one of them is broken into 5, which is the white cell. So the hematopoietic stem cell is what differentiates into all the blood cell lines, which is the progenitor. So it is the progenitor cell. Progenitor means that they all come from it. Okay, right. Someone is asking you to mute your microphone. So please keep alcohol. And if you are not speaking, please mute your microphone. Okay. There is some statistics here with this calculation that during you are supposed to mute your microphone and you are not using. Okay, I think I'm co-host, so I can mute all. All right. Let me do that from here. Very well. Okay, I've just done that. So all of you are muted by me. But if you need to speak, you can unmute yourself. Good. So in the course of producing a mature cell, the original stem cell will undergo 17 to 19.5 divisions. And each time it divides, the number doubles. So it will be 1, 2, 4, 8, 16, 32, 64, 128. That is 5 divisions, or is it 4 divisions? So after 4 or 5 divisions, it's already 128. By the time it goes through 17 to 19.5 divisions, one particular cell will give us 170,000 to 720,000 cells. That is how many cells you can get from one stem cell, which by itself also remain. So it develops into this number of cells, but the stem cell still remain. Okay. So here again, we have the table showing the multipotential hematopoietic stem cell divided into common myeloid progenitor cell or common lymphoid. Common because these cells will come from it. Okay, so natural killer cell T and B lymphocytes will come from the lymphoid cell. Plasma cell, you can meet a question that will ask you whether what is a plasma cell, or is a plasma cell a lymphocyte? So if you get a question like these cells, this cell is a lymphocyte, and then you see some other cells, including a plasma cell. I want to put on record that plasma cell is a lymphocyte. It is just a differentiated B lymphocyte that secretes antibodies. Or which of these cells secretes antibodies? And you can have B lymphocytes and plasma cell. If you have plasma cell, that is the one. It's plasma cell that will secrete antibodies. Plasma cell is a differentiated B lymphocyte that has the ability of producing antibodies. Then when you come to the myeloid, again, it's common because all these functional cells come from it. So you get the megakaryocyte. Here we see it, the megakaryocyte, giving rise to thrombocyte. You have erythrocyte, mast cell, which is a basophil, differentiated basophil, and the myeloblast. And the myeloblast will give you basophil, neutrophil, eosinophil, monocyte, and monocyte will give you a macrophage. Because macrophages are mainly in tissues and not in peripheral blood, we normally don't add them to the white cell count. Okay, so when we do the differentials, we don't add macrophage because macrophages are expected to primarily be in tissues. Their younger brother monocytes are in blood, macrophages are in tissues. And so together with the lymphocytes combination, we have 5 white cells. And
Prof.
then, hello?
Yes, Prof. Please, can you go over with the mast cell?
Okay. Mast cell is here, but actually mast cell is a differentiated basophil found in tissues. Okay, so mast cell is just like monocyte and macrophage. If you look at the picture here, you can see the mast cell and the basophil, not too much difference. So technically, we see mast cells in tissues as well. And we say that mast cells are differentiated basophils. So a basophil may differentiate into a mast cell, and then a monocyte may differentiate into a macrophage. You can see some question that says basophil is to a mast cell as monocyte is to a macrophage. Okay, so a mast cell, I will call it a big brother of basophil, a macrophage of monocyte, and both of them are found in tissues. Yeah,
okay,
go ahead.
So my question is, why is the mast cell originating from a common myeloid progenitor, but then since it's also a tissue found in tissues, but with respect to the macrophage, it's rather coming from a monocyte. So why is the mast cell coming directly from the common progenitor, but not from the basophil?
It's just the drawing here that was captured there. This is somebody's work that we try to understand. So the best from what I'm describing will be that it should be here, isn't it?
Sure.
Yes. So if it was here, that would be correct. But this is somebody's idea that I'm trying to use to teach. Is that okay?
Yes, thank you.
All right. Right, so you see some more of the cells here. This one is more expanded. But usually these are not asked in examination questions where you will be asked to find out what cells come after the myeloblast and what cells come after the promyelocytes. Is rare that something like that will happen. We normally base on the functional cells, the ones down here, and not this one. But this is to give you an idea that a neutrophil doesn't just come from the myeloid stem cell. It goes through different stages of development before it gets to a neutrophil. The same way is eosinophil and basophil. They all go through different stages. But sometimes you just draw them. If you go back here, we just say myeloblast, eosinophil. But from myeloblast to eosinophil, there are quite a number of stages of development before you get there. So here the same way, and then you get to the megakaryocyte, which is a matured end cell, break into the smaller platelet. Look at the erythrocyte. Also pronomoblast, basophilic, nomoblast, all blast. Blast means young. And then by the time you get to polychromatic erythrocyte, which is the one with the reticulocyte stage, then the nucleus is gone, extruded. Okay, so if you ask a question that do red blood cells carry or have nucleus, if it's not qualified as matured red blood cell, then yes, red blood cells have, these are all red blood cells. It's just that the functional one, we normally say red blood cell in reference to the last one, but you have to really qualify. If you are going to ask a question as to whether red blood cells have nucleus or not, then it has to be qualified of mature red blood cell. The mature one don't have nucleus. However, if there's a question like, which of these cells don't have nucleus, and you see basophil, eosinophil, monocyte, red cell, then the answer now is red cell, all right? So you have to know which of the cells they are being related to before you come into, you settle on the answer. Sometimes you have to do elimination tactics as well. Remove some, and then the one that stands, the odd one out will be it. Okay. So let me quickly go through this text here that the idea of forming the mature red cell, okay, is that the precursor cell at a particular stage is capable of following several differentiation pathways. So I'll talk about that. It doesn't jump from precursor to the final. It goes through differentiation pathways, and then it will express on its surface receptors that allow it to receive signals. So the receptor will receive the signals such as interleukin-7 or granulocyte colony stimulating factor or granulocyte monocyte colony stimulating factor, allowing the cell to become a particular cell type. The cell differentiation is thought to be a function of which cytokine it encounters. So for a cell to change into a granulocyte, if a cell, the young one, encounters GCSF or GMCSF, then if it's GMCSF, then it can be a granulocyte or a monocyte. If it's GCSF, then it can only be a granulocyte. So depending on which of the cytokines the cell is exposed to, it will become that particular cell. And this is a matter of chance. But the chance is enhanced based on the body's requirement. Based on the body's requirement, it will increase the amount of growth factor that it wants cells to become, so that the chances of they encountering it will increase. Having increased in chance, they will then produce that particular cell. So I just go back quickly and say that from here, if you want this myeloid cell to become a red cell, there are certain growth factors that are exposed to this cell and they will convert to this one. If you want it to become this, you expose them to different growth factors and this will convert to that one, all right? And depending on what you want, you increase the growth factors and that conversion will take place. Okay. So the differentiation signal is that the cell received at a particular time, say in its lifetime, is a crucial factor, the signal you receive, in determining whether it will become a white blood cell or red cell or a lymphocyte or a macrophage. The signal is what determines it. In this case, signal referring to the growth factors and then the cytokines. And here is good to mention that through a series of studies, investigators have discovered that key growth factors and cytokines that causes progenitor cells to make different type of blood cells, as some are here, okay? They were able to discover them. So you can take a stem cell and within the laboratory in a Petri dish, convert them into a particular cell line if you know the growth factors that you are supposed to add to those cells. So we know the growth factors. I don't know if there's, this table is there. So very good. It's good we have this. So look at it here. If you expose the multipotential stem cell to interleukin-3, 1,6, GM-CSF, and SCS stem cell factor, you will convert this cell into a myeloid cell. Okay? Then if you expose the myeloid cell to these factors, EPO in particular, stem cell factor is common, TPO here, interleukin-3, GM-CSF, you will convert this cell to red cell. The difference between converting a myeloid progenitor to red cell or platelets is thrombopoietin or erythropoietin. So when the body wants erythrocyte, it will produce SCSF. SCSF is stem cell factor. It will produce interleukin-3, GMCF, but it will upregulate EPO, erythropoietin. And having increased that, myeloid progenitor cell will convert into a red cell lineage. If you expose this only to GM-CSF without the other factors, myeloid progenitor cell will become a myeloblast. Expose the myeloblast to these factors and you get eosinophil. So these factors are known and you can use it as a protocol even in the laboratory when you get a stem cell, and you'll be able to convert them into a particular lineage. And this is how cells of the blood are developed, okay? You also need to see here that the factors interact with one another in complex ways. These are all by natural means, okay? And when they interact, so there are different factors, not just one. There are interaction that goes on between them and the cell, okay? And when they interact, they create a system of exquisite genetic control and coordination. And these genetic control and coordination lead to the blood cell production that we see. So the hematopoietic stem cell is a multipotent cell, that is, it has multipotential to become any of the blood cell line. So they can give rise to the cells that are more and more committed to differentiate into a given cell type. So they have potential to become red blood cells, for example, but the stem cell doesn't make hemoglobin itself. That is why you need it to become red cell, because it's hemoglobin that we need in the body, not the stem cell. If a stem cell cannot produce hemoglobin, then it must change into red blood cell that contains hemoglobin so that we get oxygenation in the tissues. That is why we need the functions. The stem cell must convert into a macrophage through the lineages to monocyte macrophage before we can get phagocytosis to protect us from infection, before we can get neutrophils, before we can get eosinophils and stuff like that. Okay. In the developing embryo, the young embryo in the body, blood formation occurs in aggregates of blood cells in the place called the yolk sac. So in the embryo, blood development begins from the yolk sac. Later, we'll talk about the fact that in the adults, the blood development is in the bone marrow. But in the embryo that hasn't got bones yet, the blood development begins in the yolk sac called blood islands. And as the development progresses, the formation occurs in the spleen, in the liver, and in the lymph node. This is very important. So in the embryo, you're going to have the blood development from yolk sac, then the spleen take over, then the liver and the lymph nodes. Then later on, when bone marrow develop, it will then assume the task of forming most of the blood cells for the entire organism, okay? So, but maturation, activation, and some differentiation of lymphoid cells occurred in secondary lymphoid organs such as the spleen, thymus, and lymph node. In children, hematopoiesis occurs in the marrow of the long bones such as the femur and the tibia. In adults, occurs mainly in the pelvis, in the cranium, in vertebrae, in the ribs, and in the sternum. This is also important. So in the embryo, we talk about the spleen, we talk about the liver, we talk about the lymph node after the yolk sac. In the adult, we are talking about the bone marrow in the pelvis, in the cranium, vertebrae, ribs, and sternum. So in some places where the red blood cell development have defect, you see the children having cranium expansion, the one we call frontal bossing. So the person's forehead will be expanded because there's the need for expansion to produce that. And so that people If you look at people from, is it Kenya and Zambia, they have some style of head, you know, heads. Here is that if the young child is not playing on pillow or something, but sometimes is from this cranium expansion that leads to those frontal bossing. So in healthy adult person, approximately 10 to the power 11 to 12 new blood cells are produced daily so that you can maintain steady levels of peripheral circulation. That is the amount of blood cells that are produced per day in an adult. Very well. Now we zoom into the individual cell development. So I'll talk quickly about stem cell maturation, then I'll talk about red blood cell, white blood cell, platelet, the individual cells and how they develop. Okay, so stem cell, we have touched on it before, that they mature and undergo changes in gene expression, and these changes limit the cell type that it can become as it moves closer to a specific cell type. I already mentioned that as the cell differentiates, they move into a different cell line and eventually become a particular cell type. So the changes can often be tracked by monitoring the presence of proteins on the surface of the cell. So when you look at the cell surface molecules, it can tell you the different stages of cell development. And each change moves the cell closer to the final cell type and further limits its potential to become a different cell type. So if the cell converts to a myeloid progenitor, it cannot change into a lymphoid progenitor. And then it's going on. If it converts into a B cell, a B lymphoid progenitor, it cannot go back to T cell, all right? And then it keeps limiting itself. So here we have some pictures to look at. This is the embryo, the 8-cell embryo after fertilization. And all the body cells and tissues would develop from here. This changes into the blastocyst, and within the blastocyst, you have the stem cell, okay? The trophoblast covers the blastocyst. If you break into it and release the stem cell, as I mentioned, you can develop all more tissues even in the laboratory. Ethics doesn't allow it, but all cells come from the cells, the amount of cells within the blastocyst, including blood cells coming out of it as well, okay? So here you have the blastocyst stage, then the inner stem cell mass. Remove these stem cell mass, and you can get blood cells, muscle cells, neural cells. This is how blood cells develop from the young, the developing embryo there. Again, out of the blastocyst, we have pluripotent inner mass. They will develop into the various organs as well as the blood cells. Okay. So about 1 in every 10 to 15,000 bone marrow cell is thought to be a stem cell. And when you look at the peripheral blood, the circulatory blood, this amount falls to 1 in 100,000. So when you make blood film, you are not likely to see a stem cell in there. The amount of stem cell in the circulatory blood is very, very minimal, 1 in 100,000 of all cells. A true stem cell is capable of self-renewal. It must be able to renew itself for the entire lifespan of the organism. What do we mean by self-renewal? If you take one stem cell, it differentiates into two. One will go on to become a neutrophil, the stem cell will remain, the other one remain. So it says that it's self-renewal. Anytime it divides and one goes off to continue to divide, one remains, and it remains to divide again and again. That's where we say stem cells do not die. Okay, and the hematopoietic stem cells are ultimately responsible for the constant renewal of blood cells, which are responsible for maintenance and immune protection of every cell type of the body. So every cell type of the body is maintained and protected. Maintained means that sustained. So if neutrophil die, it will be sustained by another neutrophil coming from the stem cell. And so your blood level cells are maintained. If you are healthy, then your blood cell level is maintained by the continuous division of new stem cells. Okay. So these are cells from a bone marrow aspirate. You see hematopoietic precursor cell. There's a promyelocyte in the center. It's a promyelocyte, and two metamyelocytes next to it. These ones. Okay. And this is a bone marrow picture, and then we have some band cells. Could be neutrophil. This could be a basophil or something. More of neutrophil. These two, and they are matured enough to be about coming out of the bone marrow into circulation. All right. Now, red blood cells, erythropoiesis, development of red blood cells. How does our body get to know that we need red blood cells? The body must have a way to assess the concentration of red blood cells in circulation so that new ones will be produced. Oops. Yeah. So new red cells will be produced to replace old and dying ones. And the regulation and production of red blood cells is called erythropoiesis. Erythropoiesis is regulation and production of red blood cells, okay? The mechanism occurs in this way. The kidney, okay, monitors the level of oxygen in the blood. I think there's a picture here. Yeah. Okay. The kidney monitors the oxygen level in the blood. If oxygen level are low, then it will secrete erythropoietin. Okay. Or there's information that goes to the kidney to say that oxygen level of blood is low. Then it means that the body needs red blood cells to boost up the oxygen level. The kidney will then secrete erythropoietin. Erythropoietin will go into the bloodstream and travel through the body. All cells will be exposed to it. But guess what? They don't have the receptor for erythropoietin if they are not going to be red cells. So only red bone marrow cells, the erythrocytic erythroblast, the young erythroblast is called best forming unit erythroid. The young, very, very young red cells, they have receptors for erythropoietin. So they will pick up the erythropoietin and then they will respond to it. The erythropoietin will stimulate the production of these erythrocytes in the bone marrow. They will then leave the bone marrow, move into the bloodstream, and as their population increases in the bloodstream, the oxygen carrying capacity of the blood will also go up. When the kidney senses that the oxygen level is enough, they will respond by slowing down the secretion of the erythropoietin, which in this case serves as a negative feedback. And then the red cell level will maintain. This cycle will continue on and on. So the negative feedback ensures that the size of the erythrocyte population remains constant. Is red cell excessively. Okay, so your concentration level is abnormal because you continue to keep producing them. So here we have it in pictures. That is the kidney receiving this, the signal. So it senses that RBC level is low. It releases erythropoietin, which travels to the bone marrow. and then instigate the production of the red cells. They then move into circulation. The level of oxygen is okay. There's no more signal coming. This release is stopped. And then the cycle goes on and on and on. Okay? That is how red cells receive the signal for production. And the developmental stage of the red cell starts with colony forming units, granulocyte, erythrocyte, monocyte, and megakaryocyte. So this is CFUGEMM is colony forming unit, granulocyte, erythrocyte, monocyte, megakaryocyte, meaning that this cell can be a granulocyte, it can be a erythrocyte, it can be a monocyte, it can be a megakaryocyte. That is what it means. And it has stem cell factor CD33 and 34. When this cell is exposed to stem cell factor and interleukin-3, it changes into best forming unit erythroid, BFU, burst, BURST, forming unit erythroid. Okay? And when the best forming unit erythroid, not BEST, is BURST, burst. When this best forming unit erythroid encounters IL-3, SC factor, and EPO erythropoietin, it changes into colony forming unit erythroid. Further exposure to erythropoietin, it converts into erythroblast. More exposure to erythropoietin, it converts into pro-erythroblast and then convert into erythroblasts that have glycoprotein A on it. More exposure to erythropoietin, the erythroblast changes into reticulocytes. So you can see the importance of erythropoietin in the developmental stages of red blood cells. And finally, reticulocyte changes into erythrocytes, the final functional part of the red blood cell. This is the red blood cell developmental stages as they go through from the stem cell all the way to the erythroblast. So you still have it here. Hello?
Yes, please, can you go back to the previous slide?
Here?
Okay, so the best forming unit, the colony forming unit, do they also have the receptors for the erythropoietin, or it's only the best forming unit?
It starts from here and not there. So best forming unit has a receptor for erythropoietin, yes.
Okay.
But this
And then the colony
forming unit GEMM has receptors for IL-3 and SCF.
Okay, so does it mean it's only the best forming unit that has the receptor for the erythropoietin, apart from the EFUE? Those ones do not have receptors for it.
You mean this one?
Yes.
Can you see the cursor? This it has. That is why it's exposed to EPO. It does have. From here,
okay,
all these stages are exposed to EPO, meaning they have receptor for EPO.
Okay.
From there, from this stage, yeah. Okay.
Okay.
And if this cell was exposed to GCSF, it will change into a granulocyte. Here.
Okay.
A granulocyte. Yeah. Is that okay?
Yes. Yes.
Okay. So erythroid lineage, again, stem cell to best forming unit erythroid, colony forming unit erythroid, pro-erythroblast. You see here, we are even adding more. Basophilic erythroblast. It wasn't in the other one. And at that stage, you look at how the neutrophil nucleus is about to come out. So at the reticulocyte stage, the developing red cell doesn't have nucleus. They only have remnant. So the nucleus is lost at the autochromatic erythroblast. Okay. Folks, this is important. Let me just join you.
Hello, everybody. It looks like my audio is not playing, or is it across board?
Mine is also not playing. I thought it was my network.
I think
Folks,
you
I'm the
received
one who
the
is
call.
not talking. Oh, I'll come to you in a minute.
Oh,
Okay.
okay.
All right. To speak to him. Whenever your boss calls you, whatever you are doing, stop and talk to him. Your job depends on it. So I know you forgive me. All right, those of you who couldn't hear, like I said, it was me that went off. I am back. Right. So here, by the autochromatic erythroblast stage, the nucleus is lost, and then we go to the reticulocyte. It has a little bit of RNA remnant material, so it stays different, and then it converts to erythrocyte. When there's anemia, severe anemia or bleeding or hemolysis, you will find reticulocyte in circulation because the circulatory blood is calling for cells from the bone marrow, and mature ones are not enough, so they are joined by reticulocytes. That is why we do reticulocyte count in diagnosing severe anemia or acute anemia situations. So when you see reticulocyte, it shows that there is some crisis and all cells are required. For example, if I go to laboratories in hospitals and I find laboratory students and not qualified ones in the lab, it will tell me that there's some kind of situation going on. Maybe the qualified ones are on strike and students have been called in to come and take over. And so counting for reticulocytes is indicative of the fact that you don't have enough cells, mature red cells around. Okay. So here we have it in proper pictures. You could see them well. Basophilic erythroblast is looking beautiful here, isn't it? And then under the microscope, you see the right one there. Oh, can't deal with this. Chromatophilic erythroblast, you see them there. Normoblast, and then reticulocyte. And this is reticulocyte, right? This is the normoblast, okay? And that is erythroblast from blood film. So when you do this is a smear from bone marrow, looking at developing red blood cells. So the black arrow is basophilic erythroblast. There you are, and there. And then the green is polychromatophilic erythroblast. There you go. Okay. Obviously, if this was shown to you in an exam, I'm not really sure you'll be able to identify them. So we won't be bringing something like this, but it's good for your education. Okay. We move to white blood cells. We have 5 types in here: neutrophil, eosinophil, basophil, lymphocytes, and monocytes. So development of white cells, we call it myelopoiesis. Myelopoiesis is the regulated formation of myeloid cells, including granulocytes, macrophages, mast cells, and dendritic cells. Okay, and then the granulocytes are also referred to as polymorphonuclear leukocytes. And this is because of their oddly shaped nuclei. Polymorph. Poly means more than one. Morph refers to morphology, and then nucleus. So it means that this cell have more than one nuclei. Okay, and they give rise to 3 short-lived cell types. These are eosinophil, basophil, and neutrophil. The other white cells, monocytes and lymphocytes, are not polymorph. They have nucleus or nuclei, but it is one whole nucleus. So that is mononuclear cells. So the white cells can also be divided into polymorphonuclear cells and mononuclear cells. Mononuclear is monocytes and lymphocytes. Polynuclear is eosinophil, basophil, and neutrophil. Then we have the ones that have granules and the ones without granules. So the polymorph usually have granules. Being polymorphonuclei, they are granulocytes. So they differentiate into the different types in process called granulopoiesis. So when you say granulopoiesis, you have limited the development into granulocytes. So granulopoiesis is the regulation and the development of the white cells that have granules. And these are the 3 polymorphs. So they first transform from a common myeloblast, myeloid progenitor, to a common promyelocyte. Don't worry about this one's myeloblast, myelocyte, and the which follows which is not really important in examination. Promyelocyte give rise to a unique myelocyte that for the first time can be classified as eosinophil, basophil, or neutrophil progenitor based on the histological staining affinity. If you know how the cells are named, eosinophil, eosin larvae cell; basophil, basic stain larvae cell; neutrophil, neutral staining cell. So when you put Leishman stain on a white cell, the eosinophilic cells will pick the eosin and not the methylene blue. The basophilic cells will pick the methylene blue and not the eosin. The neutral cells will not pick either. So the granules of neutrophils are not picking any stain, whilst the granules of eosinophil pick eosin, and the granules of basophil pick the methylene blue. And that is how we're able to differentiate the 3 cells: eosin larvae, basic stain larvae, and neutral larvae cell: neutrophil, eosinophil, basophil. Okay. So here the unique myelocyte differentiate into metamyelocyte and then a band cell with a C-shaped nucleus before becoming a mature eosinophil, basophil, or neutrophil. Macrophages come from monoblasts, progenitors that differentiate into promonocyte, which mature into monocytes. Monocytes eventually enter into tissues, remember that, and become macrophages. When monocytes go into tissues, they differentiate to become macrophages. So macrophages are not entirely a different cell. They are only differentiated monocytes. You have granulopoiesis here, promyelocyte, basophilic metamyelocyte, basophilic myelocyte, neutrophilic, eosinophilic myelocyte. That's the myelocyte stage. then the metamyelocyte stage, the bind stage, and the final cell, basophil, neutrophil, eosin. Look at the eosin color, medium blue color, and none of the colors standing out for the neutrophil. So the neutrophil granules don't pick either of the colors, okay? Or they pick the sheen of the combination of it. But that is the basophil, right? Good. Here, the smear is showing developing neutrophil. So neutrophilic myelocyte, early neutrophilic metamyelocyte, and late neutrophilic metamyelocyte or stub cell over there. Okay, very good. So, thought I could have platelets for you, but it's not there. So if I want to talk a little on the platelet side, then it's just that the cell will develop into the megakaryocyte and then it will break off into the smaller versions of platelet. Do platelets have nucleus? Do platelets have cell membrane? Yes, they do, okay? So when they break off, they have a membrane covering them, even though they are that small. They have nuclear material inside as well that keeps them. And then they have granules that contain the components that they use. All right. So we've done. If you don't have any question, we've just about done an hour, and so we have the opportunity to do one more for today. And as I said, I will do the RBC morphology. So I'll stop screen sharing. If you have a question at this point, otherwise I'll switch over the slides and we will have a look at some of the morphological appearance of some of the cells during pathological states. We have information, a little bit of information on deficiency anemia as well as hemolytic anemia, and then red blood cell morphology. So let me start with the red cell morphology then. If you don't have a question. All right, here you go. Or do you need a break? Do you need like 5 minutes break? I need to ask.
Yes, maybe some 5-minute break.
Hello?
5 minutes.
Please continue.
Okay.
Hello, Prof.
Yeah.
Some 5 minutes break will do.
Okay. Okay.
Thank you.
Yeah. So 5 minutes.
5 minutes.
Okay.
5 minutes. So 5 minute break.
And he's teaching too.
You can't hear.
He's teaching.
And the sound is not coming.
This
No, I am not teaching. We just had a 5 minutes break, so I'm just coming back.
But I'm not hearing it in the mic.
Can you hear me?
Your battery is also low.
Hello. I can hear you. We are just about starting.
Yes, we are on.
Okay.
Okay.
All
All right.
or when you say you can't hear yourself out.
All right. So this one is morphological changes in diseases, how the red blood cell shapes change during some disease conditions. So I'm going to go through the different changes, okay, that we have. Okay, so we start with the sickle cell. So we see sickle shaped red cell. I'm sure you may have seen, majority of you have seen this shape before. So you can get a film like that, and then the arrow goes and you're asked to indicate the type of cell that the arrow is hitting, and maybe you have options. In here, you can see quite a number of different cells. Here you see something like a chinocyte. This is like a target cell or so. but the key point will be like that. And then the center pillar of red cell. The center has no hemoglobin at all. So in iron deficiency, you see some cells like this a lot. And this is about a normal red cell and that one. Red cells normally have a little bit of pillar in the center because of the shape. Okay, you can't have it all filled up. That is when you have like this. Is not normally what we see. Okay, so this is found in hemoglobin S disease. Then we have hypochromatic red cell seen in thalassemia. So the center is larger. So it is hypochromatic because the staining is less, hypo. Why is the staining less? Is the hemoglobin in the red cell that pick the stain, making it more prominent. So if the hemoglobin is less in the red cell, then it will stain hypo, okay? And so hypostaining is related to the hemoglobin content, which is what picks the stain. So if you look here, the center pillar is very large. For these ones, the center pillar is very, very large. There's bigger space, meaning the hemoglobin is just around the size, the edges of the cell. And we see hypochromic or hypochromatic cells in thalassemia cases. Then we have spherocytosis. The red cells are more spherical. Okay, it doesn't have the normal red cell shape, that is the oblong nature of it, making the center a little bit spreading. So if you look at this one here, that one there, this is very rounded, 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 rounded. All these are circular cells, very rounded, spherical cells. So here you see spherocytosis. So the red cells are more spherical. They lack the central area of pillar. There's one here, if you look here, you can see a little bit of center pillar, and this is not spherical. So this is a normal, almost normal red cell, as compared to maybe that one and that one that don't have the center pillar at all. So when you don't see the center pillar, which is the paling, then you are looking at spherocytosis. This is found in hereditary spherocytosis, immune hemolytic anemia, Zeev syndrome, macroangiopathic hemolytic anemia. You will see spherocytosis. Okay. Then the reason why they have formed that shape is because of deficiency in anchoring and spectrin. I don't want to waste too much of your time here, but the cytoskeleton is what holds the cell up in its set structure or stature. So the cell is held up, the cytoskeleton, which is made up of anchoring and spectrin proteins. So when the anchoring and spectrin molecules are deficient, the cell loses some of its structure and rather becomes spherical instead of the oblong nature of it. It leads to release of macrovesicles as well. The released macrovesicles causes the cell, this is the normal one, to become more rounded. And that is what we see in spherocytosis. Okay. Then we have elliptocytosis. The cells are elliptical. They say they are oval or elliptical in shape. Long axis is twice the short axis. This is the long axis and is twice as long as the short axis. This here as compared to that. So the long axis is twice the size of the short axis. So these ones here are the elliptocytes. They are found in hereditary elliptocytosis, megaloblastic anemia, That is B12 deficiency anemia, iron deficiency anemia, thalassemia, and myelofibrosis. They are found in there. Then we have oval cells, ovalocytes, with elongate pale stoma. Okay, so you see the stoma in there. That is the stoma. Okay, round there is here, there, this ones. Okay, they form the stoma, and this is found in hemolytic anemia, thalassemias, and hereditary erythrocytosis. We find ovalocytes in there. Then we have stomatocytosis, stomatocytes. These are red cells of a central linear slit or stoma. Again, there, slit across the center. Instead of the center pillar, all you see is those ones there across. So it's seen as muff-shaped form in peripheral smear. It's like a muff. Maybe those ones there, here. This is not classical, yeah, that one there. Okay, some in here. So seen as muff-shaped form in peripheral blood smear, found in alcoholics, alcoholic liver disease, hereditary spherocytosis, hereditary stomatocytosis, including some rare patients of altered expression of RHAG and RH30, and then an epitope. This is an epitope on a subtype variant of rhizome antigen. Okay, so people with that epitopes end up with stomatocytosis. Then we look at variation in size, shape, or staining in red blood cells. So this is anisocytosis. Anisocytosis is increase in the, you're going, okay. Increase in the variability of red cell size. So in one film, you see a large cell, medium cell, small cell. There is variation in the sizes of the cell. That is anisocytosis. So variation in erythrocyte size is now measured by the red cell distribution. So when you do full blood count on the ultra analyzer, RDW is what measures the anisocytosis. So the RDW should be taken into account when interpreting the mean corpuscular volume, the MCV. In this situation, I want you to think about how the MCV will look like. In anisocytosis, what do you see with the MCV? I'm not going to ask you to tell me, but just note it and look out for it. What is the MCV like in anisocytosis? Okay, when the cells are varied in size. Then we have teardrop cells, red cell shaped like a teardrop or pear shape. That one. Okay, yeah, like papaya kind of thing. It's found in bone marrow fibrosis, megaloblastic anemia, iron deficiency anemia, and thalassemia. And then we have pencil cells, red cell shaped like a pencil or cigar. 1, 2, 3. Found in iron deficiency anemia. We also have macrocytosis. Here the red cells are large, they are increasing size. So in folate and B12 deficiency anemia, we see macrocytes. And so the red cells are quite large, but their number may not be that much. So the red cells are larger than 9 microns in diameter, maybe round or oval in shape, the diagnosis significance being different, found in folate and B12 deficiency, alcoholism, liver disease, and reticulocytosis. Then the microcytes is not a classical film for microcytes, but why we have to note in macrocytes is that the red cells are decreased in size. They are smaller than 7 microns in diameter. The nucleus of the small lymphocyte, they look like the nucleus of a small lymphocyte, okay. And it's a useful guide to the size of a red cell. So the nucleus of a small lymphocyte can guide you. They are found in iron deficiency anemia, mainly thalassemia, sideroblastic anemia, lead poisoning, anemia of chronic diseases. You see microcytosis. Here also, I want you to look at relation with MCV. In fact, macrocytes, microcytes, and all that, try to relate it to the MCV. How would the MCV look like in the situation of microcytosis or macrocytosis? Acanthocytes, spherical cells with 2 to 20 spicules. So the spicules are on the side of it, 2 to 20 of them. Acanthocytes of unequal length and distributed unevenly over the red cell surface. Is found in liver disease, post-splenectomy, anorexia nervosa, those who don't eat by choice, they don't eat by choice. By starvation, usually is not by choice. Okay, we see acanthocytes. Then we have echinocytes. This one is seen very, very frequent. When you leave blood to stay on the table for a while before you make a film, you tend to see echinocytes. They are covered with 10 to 30 short sparkles of regular form. They are found in uremia, severe burns, EDT artifacts, and liver disease. You should add that films that are not made in time, you sometimes see that. Then schistocytosis is fragment of red cells, fragmentation. You see hemolytic anemia, yep, the red cells are hemolyzing. Macroangiopathy, DIC, disseminated intravascular coagulation, and mechanical hemolytic anemia. You see fragments of red blood cells. Okay, schistocytes, fragments of red blood cells are known as schistocytes. Then the target cells, you have the targets. So there is no hemoglobin, and then in the middle somewhere there is a stash of it. So there's a target in the middle of the red cell, target cell. Red cell have an area of increased staining which appears in the area of central pillar. So the increased staining is in the area of central pillar, is in the middle there. Found in obstructive liver disease, severe iron deficiency, thalassemia, hemoglobinopathies, hemoglobinopathy, that is the sickle cell disease, SNC, and post-splenectomy. Then we have Howard-Jolly body, small round cytoplasmic red cell inclusion with same staining characteristics as the nucleus. There you have it. There. Okay, found in hemolytic anemia, post-splenectomy, and megaloblastic anemia. Then we have Rolio formation. Okay, this also yes seen in old blood. The cells are stuck to each other. So hyperglobulinemia, hyperfibrinogenemia, you see that? Stacks of RBC resembling a stack of coins. That is the Rolio. Red cell agglutination, there's irregular clumps of the red cell. So the red cells are clumped together. We see it in cold agglutinins, warm autoimmune hemolysis as well. Phagocytosed red blood cell. This red blood cell is found in the macrophage. So that is a macrophage, and the red cell is in the middle of it. It's So red cell in vacuum is cytoplasm of neutrophil, or this one is a neutrophil. So here. So that is the neutrophilic, the polymorphic nucleus here. And then the red cell is in there. Found in infection. So during infection, the cells are phagocytosing infectious agent, and they end up phagocytosing red cell as well. Autoimmune hemolytic anemia, incompatible blood cell transfusion. So when you're doing transfusion investigation, transfusion reaction investigation, you can make a film and see if you will see phagocytosed red blood cell. Then we have polychromasia, red cell stain shades of blue-gray as consequence of uptake of both eosin by hemoglobin and basic dyes by residual ribosomal RNA. So often slightly larger than normal red cells are round in shape, round macrocytosis, meaning that the red cell didn't lose all the RNA material. So it's bordering on mature cell and has reticulocyte tendencies, okay? So they are not typical, they are not your typical reticulocyte, neither have they converted fully into a mature red cell. Sometimes you see conversion of reticulocyte into mature red cell in the peripheral blood. That can happen. And in situation like that, you see polychromasia. So you see it's a situation with reticulocytosis. When the reticulocytes are more, you see polychromasia. So for example, bleeding, hemolysis, or response to hematinic factor. If somebody is given blood tonic, which in this case called hematinic, and the blood tonic will instigate production of red cells. But if there aren't enough mature red cells in the bone marrow, then the bone marrow will be forced to ship out immature cells to compensate for the demand. And you will see polychromatic cells in there. This is good old malaria parasites in red blood cell. These are red cell inclusions as well. So it's important that in hematology, the problem with malaria is whether it belongs to hematology or parasitology. We're usually not sure, but the jury is out on that. Each of the disciplines will want to claim. Hematology will claim it as a red cell inclusion. Parasitology will claim it as part of the parasite that they look at. This is typical Plasmodium falciparum in there. You see it close up here. Yes, sir.
Please, is there no audio? I'm the only one not hearing the audio.
not hearing him. I'm sure he's preparing for us.
Is it
I
when
think
I
he-
just came back, or all along you didn't hear me?
We didn't hear you all along. The last thing I heard was the malaria parasites.
Yeah, so my network went off and then I came back. I think when I just joined back, the microphones was off. So I started from here, right? This is why you were not hearing from me. But prior to that, you could hear all this side, right?
Yes, please.
So just before I ended, my internet reset. So I was here, and then I said, we have seen the gametocide, and then I said that is the end. So what I was saying was that we have done this in 20 minutes, very good time. We still have about half an hour of the allotted time to this course. So what is here? So you think the lecture is complex and bulky? Wow. Okay. Latif, there's a conversation in the chat, so if you want to have a look at it.
Okay, I'm looking at it.
Okay. All right. Anybody with a question?
Hello, Prof.
Hello?
Are we able to share the slides with them?
Latif?
Hello?
Yes. Were you saying something?
I was asking if we can share the slides with them.
The lecture slides?
Yes.
Yeah, I can. I thought you were recording, were you?
Yes, we are recording a video, but video will be bulk to share on
bulky. Yeah.
WhatsApp. Yes.
Yeah. I don't mind. I can give it out.
Okay.
So
all right, that's fine. So that those who are not here or those who are here will get the benefit of the explanations and all that.
Yes.
So if you are not here, you are just taking the slides.
Sure.
You get everything. I think it's okay.
Yeah, so what I'll do is that the ones we finished with, I will send to you and then you can distribute.
Okay.
Yeah, it's okay with me. I don't have it. Okay. All right. So can I have another go at something else? Okay.
Prof, it appears there are no questions, but we'll still keep our platform open. Anybody who has a question can put on the platform to be forwarded to you.
Yeah, and then at any time,
the next
point,
you can address those questions.
Sure. Okay. So then let's do a bit of deficiency anemia. See how much we can utilize the time. About 23 minutes to go. So let me check my microphone is on. Right. All right. Okay. So deficiency anemia. We want to define anemia first. we want to define anemia first. And anemia is operationally defined as a reduction in one or more of the major red blood cell measurements. So if, for example, we have less hemoglobin, but normal hematocrit or normal red cell count, it will still be considered as a presence of anemia. If we have less hematocrit and normal hemoglobin or RBC count, it can be determined as anemia based on the reference range that we have for the individual measurements. The good news is that most often than not, they tend to collaborate each other. But for example, you can have macrocytes, and the macrocyte count will be low, but hemoglobin may be normal. And in that situation, you still can determine that anemia exists. The hemoglobin is normal because there's enough of them in the few cells that are existing. So says, remember that these measurements are concentration measures. And so you tend to sort of quantify them. Okay, they are quantifiable, but a reduction in any of them could result in anemia. So the anemia is also defined as a condition where the total number of red cells is reduced or the cells have a decreased hemoglobin content. So you have less than 11 grams hemoglobin per 100 centimeter cube of blood, or less than 4 times 10 to the power 12 cells per decimeter cube of blood. So it can be absolute or relative. And the relative is that, as I mentioned, if hemoglobin is less in relation to the other parameters, then you have relative deficiency. If both parameters or all three are less, then the anemia is said to be absolute, okay? So it's absolute when all parameters sort of inform the anemia, or if one is less and the others are normal, then we are looking at relative deficiency. So causes of the decrease will be the fact that RBC numbers are not being produced adequately, or there's production of abnormal cells or hemoglobin, or there's increased loss of red blood cells. These are the three main causes of the loss or the causes of anemia. There's decreased production, so the number of red cells are not being produced. There's production, but the cells being produced are abnormal, or the hemoglobin being synthesized are abnormal. Then there's loss of red blood cells more than normal. These can cause a decrease. So what is therefore is deficiency anemia? They are the anemia caused by the decrease or deficiency of components that are needed to produce adequate and functional red blood cells. Functional meaning normal red blood cells. So when there is a decrease in the components that you need to produce adequate and functional red blood cells. So for example, if you need folate and there's a decrease in folate, then we are looking at folate deficiency anemia. Vitamin B12, B12 deficiency anemia, erythropoietin deficiency anemia. Things like that. So what you need and to produce the cells and you don't have, then you are looking at anemia of those. So this is in effect leads to reduction in the total number of red cells or the cells have a decreased hemoglobin content, resulting in absolute or relative hemoglobin deficiency. I've mentioned what the absolute refers to and relative. When all the parameters are missing or are reduced, then the anemia that occurs is absolute. When one is reduced in relation to the other, then the anemia that ensues is relative. Okay. So how do we evaluate anemia? First, you look at the history. So you check genetics or presence of a disease, and then symptoms. If you look at the arrow here, we have clinical assessment. That is where we do the history thing. Then we have clinical and laboratory data analysis, and we have laboratory investigation. Physical examination and science also forms under the clinical assessment. Then we have the laboratory testing. When we have done the laboratory test or investigation, you combine the clinical history and the laboratory data, and then you determine the anemia. So clinical assessment, followed by laboratory investigation, followed by the combination of the two, we arrived at diagnosis of anemia. This is indicative of the importance of medical laboratory science in this situation. Okay, so we look into the laboratory process by which we can determine this anemia. We first of all will do full blood count or complete blood count and look at the indices such as the hemoglobin, hematocrit, red cell distribution width. Then we do blood smear and study the morphology. Are we seeing anisocytosis or poikilocytosis, or are we seeing polychromasia or microcytosis? They are all important in supporting the results that we get for the full blood count. And then there's less common test, which is depend on history. We will do bone marrow, serum iron analysis, usually is serum ferritin, and then total iron binding capacity. These are all done to look for the existence of anemia. Ferritin is done in hematology. It's also done in chemical pathology. Chemistry people also do ferritin. But total iron binding capacity, I believe, is hematology. These are some of the tests that are done in diagnosing anemia. So how do you classify anemia using morphological? So first, you measure the size of the red blood cells. And by calculation from independently measured red blood cell count and hematocrit, MCV is obtained. Now you have machines that will do these things for you, but MCV, femtoliter, is measured in femtoliters, right? And is equal to 10 times the hematocrit, which is in percentage, divided by red blood cells times 12 to the power per liter. So when you do this calculation, you get the MCV. But currently, because we have machines giving us the results straight away, we tend not to be bothered too much about the formula or formulae for calculating these things. The MCV will give you the mean cell volume. So you give you the mean cell volume of the individual cells. It will count all of them and take the mean for you. Okay, so to classify anemia morphologically, you will have it as microcytic, that is smaller cells type of anemia. So the cells are microcytes or tiny. Pneumocytic, the cells are normal, but there is still anemia. And macrocytic, the cells are larger than normal, but there is still anemia. So we have microcytic anemia, pneumocytic anemia, macrocytic anemia. You can throw in the staining, and then you have microcytic hypochromic nomocytic hypochromic or nomocytic nomochromic, macrocytic hypochromic, meaning size and staining. So how the cells look like in terms of size and how they have stain. So the chromasia is the staining characteristics. Then the cytic is the size. In fact, cytic is actually cell. So micro is small, is normal, and macro is larger. So here you go, you have macrocytic hypochromic anemia from iron deficiency anemia, meaning the cells are small, and then we have hypochromasia. So the center pillar is larger, even though the cells are small. They are not picking the stains. Then we have microcytic nomochromic anemia. Here can be an erythropoietin deficiency from kidney failure. The cells are small, but the staining is normal. Macrocytic nomochromic, the cells are larger, but the staining is normal. Could be folate deficiency or vitamin B12 deficiency. Then we have nomocytic nomochromic, NCNC. This is normally from blood loss, prosthetic heart valves. When they put metal clip in the heart valve to open the heart up, they tend to break down red blood cells, and we see nomocytic nomochromic anemia. Sepsis, infection, tumor, or aplastic anemia, where all the cell numbers are reduced in this production. You find nomocytic nomochromic. So here, normal cell size, normal staining, but you see that a count will be affected. So RBC count may be reduced in this situation. Okay, what is the etiologies of this anemia? Production defect, we are looking at nutritional deficiencies. So vitamin B12, folate, or iron deficiency. We can also see anemia in inflammation or in chronic diseases. Anemia of chronic diseases, normally chronic kidney disease, you will find anemia there. Primary marrow disorders. So the bone marrow has a problem. Pure red cell aplasia is not producing any red cell at all or very little. And then myelodysplasia is not producing cells of the myeloid lineage. So myelodysplastic syndrome. The cells of the myeloid lineage are not being produced in adequate numbers. Or instead of all the myeloid lineage cells that affect red cell as well, you can have pure red cell aplasia. The lack of production is now purely linked to red cell. Then sequestration, hypersplenism. The spleen keeps some amount of red cell, okay? And they are keeping more than necessary. So the overactive spleen is removing more red cells than they should. When red cells are deformed, like in sickle cell stage, they get into the spleen and they are removed by macrophages within the spleen. If the activity continues and is extended to even normal red cells, then that is going to affect the red cell numbers. And of course, we will see anemia. And this anemia is with mild pancytopenia, meaning the other cell lines number also reduces a little bit. Then we have dilutional. Anemia affected by dilution, that is a patient's plasma volume increases with laying down and when they quit smoking, for example. So this is possibly responsible for as much as 3 to 6% drop in hematocrit in the first 2 days of hospitalization. When a patient, for example, is hospitalized, you may end up seeing dilutional anemia. It's because they are accumulating a lot of fluid and their blood volume is more diluted. So plasma level increases and it will affect the red cell. But this will correct as soon as they become active. Then we have blood loss, hemorrhage, rupture of veins and stuff, and then blood is oozing out. When we lose blood, it can result in anemia. Blood destruction, hemolysis, breakdown of red cells, excess breakdown of red blood cells also will lead to anemia. Erythropoiesis affect the number and quality of the red cell. Number will lead to anemia, quality will still lead to anemia. So two main deficiencies that we will talk in detail: nutritional deficiencies and hormonal deficiencies. Okay, so this is a typical iron deficiency anemia. You see microcytes, and this is macrocytic hypochromic picture. The cells are small, and then the staining, the center pillar is so large. So you only have hemoglobin at the edge of it. So this picture, as soon as you see this, you should know that we are looking at macrocytic hypochromic anemia, typical of iron deficiency anemia picture. Okay. So we can look at a deficiency in terms of decreased red cell production. What will cause red cell production to be decreased? And this decrease results in aplastic anemia. So when the bone marrow is defective, it fails to produce all the blood cell lines in sufficient amount. So we see pancytopenia. Pancytopenia is when all the blood cell lines are not produced in sufficient amount, including red blood cells. And so red blood cell, lack of it lead to anemia, and the anemia that results is known as aplastic anemia. So aplastic anemia may be inherited. We see it in Fanconi syndrome. And this is due to an autosomal recessive mutation. So there's lack of DNA repair in this individual. And so synthesis of the hemoglobin and the development of red cells is affected, leading to lack of production of proper red blood cells. Then aplastic anemia may also be acquired, and this is by cytotoxic drugs. There are certain medications that you take that leads to breakdown of red cells. So we see aplastic anemia, the ionizing radiations that can break down red blood cells, xenobiotics, example DDT, infections, viral hepatitis also lead to aplastic anemia, or can be secondary to marrow infiltration by cancer, example lymphoma, can lead to anemia as well. Various causes of anemia that we look at. So in aplastic anemia, we have the hemopoietic elements in this bone marrow biopsy are markedly reduced. We don't see too much of hematopoietic elements in this. All these sides are cleared up. There's nothing there to indicate cell production. Here you see a few cells in there, but majority of this bone marrow vasculature shows no cells. Okay, so in aplastic anemia, RBCs and platelet and granulocyte will be diminished, and the cause may be a drug causing or unknown cause. So there's different ways of looking diagnosing or establishing the presence of anemia, including bone marrow biopsy, as we can see here. Okay, here we are looking pretty much at microcytosis. The red cells are larger, the very little center pillar, and then we can also see some ovulocytes in here. Okay, so we probably are looking at macrocytic anemia if it exists in this film, this morphological blood film. Okay, so how do we produce abnormal red cells? In terms of the larger red cells, megaloblastic anemia, what happens? Is here due to defective metabolism as a result of reduced DNA synthesis. When the cell is going through developmental stage G1, GO is when they go into senescence, they come out into circulation. We have G1, then the synthesis stage, and G2, and then you have the mitotic stage or mitosis. So, and then the cell divides into two, and you have normal red cell. Okay, so you have the normal red cell here, and then the DNA synthesis fail. So earlier I talked about the factors that are required. Folate and vitamin B12 are needed for the at the stage to the cell growth gets stuck here. So it doesn't B12 deficiency state. And this is the B12 deficiency could be as a result of lack of intrinsic factor produced by the stomach parietal cells, okay? And if intrinsic factor is not available, your body will not be able to absorb vitamin B12, and then it will lead to lack of synthesis because it's required for DNA synthesis. And if the DNA synthesis is not done, the cell cannot move to the next stage, let alone divide into the two daughter cells. So you have it stuck here and becomes larger than normal, okay? Okay. So we also have autoimmune antibodies. Okay, this is why we don't have the intrinsic factor. When antibodies attack the gastric parietal cell, the parietal cells are the ones that produce the intrinsic factor. So in autoimmune situation where the stomach parietal cells are destroyed, there will be no release of intrinsic factor. Therefore, no binding of vitamin B12. And so RBCs become macrocytic. They are immature, they have short half-life, and they may be hypochromic as well. Neutrophils in this situation are hypersegmented. So we have B12-dependent megaloblastic anemia referred to as pernicious anemia. If the megaloblastic anemia is due to B12 deficiency only, it is also referred to as pernicious anemia. But in some cases, you will hear that pernicious anemia and megaloblastic anemia are the same. They are interchangeable in some books, okay? But pernicious anemia or megaloblastic anemia is referred to as pernicious anemia if only the megaloblastosis is dependent on deficiency, B12 deficiency only. So here we go again, macrovasculitis and hypersegmented neutrophil supporting pernicious anemia. But you have a little bit of center pillar in these few cells there. All right, it's past 6 o'clock, so I'm looking for a good place to end this one. And the house. Okay, let's do those 2 slides and then we end there. So defects in HB synthesis is also there, and here that is hemoglobin synthesis. So iron deficiency anemia is linked to defect in hemoglobin synthesis because you need iron to produce the heme that combines with globin to form hemoglobin. So if you don't have iron, the iron due to low dietary iron absorption syndrome and inadequate iron stores, you will not form the heme, and so forming hemoglobin becomes a problem. And that is why in iron deficiency, you see less hemoglobin. And if the hemoglobins aren't more, the cell sizes tend to be smaller as well. And we have glucose 6-phosphate dehydrogenase deficiency. And this is due to failure to maintain the NAD pH and reduce glutathione pools. So it reduces biosynthesis and redox state generally, leading to fragile cells that can hemolyze. And so when the cells hemolyze, you will see that anemia ensue, and it is because of G6PD deficiency. In hemolytic anemia, you might mention G6 deficiency there also, because the end result is anemia due to hemolysis of red cell. But when we are talking about deficiency anemia, you cannot leave out G6PD. So you have hypochromic cells. Look at the cells here. Very large center pillar in here, only small amount of hemoglobin around it. These are hypochromics. All right. So let me end here. Anytime we get together again, I will check the calendar, and I'm sure coordinator will keep me informed when our next meeting is. We will start from here and continue to the end and do the other ones that we have to do to complete this program. So thank you for showing up and listening to me. If you have any question, even though it's time, I wouldn't mind answering. If you don't, we will end, or my section will end now. Okay.
Thank you so much, Prof. So the floor is open for questions. If you have any question now, you can drop it. If not, and you want to send it later, you are free to do so. So we forward it to Prof to address them in our next lecture. Okay, Prof, there's a question here. I don't know whether it's been addressed. Somebody is asking, which is a good measure of chromasia from the FBC results, MCHC or MCH?
Well, when you say MCHC is looking at the individual cell, and then the MCHC, then MCH is the mean corpuscular hemoglobin. That is the average amount in each of the red cell, average amount of hemoglobin in each of the red cell, all right? So the MCH talks about the general one, and then so he says to measure what? Was it in the chat? Let me see.
Chromasia. Hello, Prof.
Yeah, I'm looking at it. Yeah.
Okay. Yeah.
Yeah. Well, in this case, we're looking at the measurement, the indication of the colors, right? So and the amount of hemoglobin will lead up to the how deep, if you like, how deep the cell will pick the color. So in this case, we then have to look at it individually. And I don't have ready-made answer, but I will go for MCHC, which look at individual concentration of hemoglobin in each cell. Okay, because you cannot measure the chromasia as a bulk. You are looking at it. It's not a straightforward linkage aside, but what I would say is that when you are looking at how the cells will pick the color, then you are looking at, you can link it to the amount of hemoglobin. For example, if you are still seeing my screen, let me go back here. Not this one. So this is chromasia, okay? So the amount of hemoglobin in the cell indicates how the color has been picked, or the deepening of the color. Now, the deepening of the individual colors cannot be quantified. So you have to look at it individually. And so MCHC, which look at the mean concentration of individual cells, will be more indicative of how the cells pick the color more than the one that combines the MCH, which is a combination of the concentration, the mean concentration of all the cells combined. So even though there isn't a ready-made linkage, if we have to think on our feet, I will go for MCHC, which is linked to individual concentration, and then the individual cells will pick the colors differently. And so that links it better than if you were to look at MCH. As I said, because you cannot quantify the chromasia. So there isn't anything like a mean chromasia, but the chromasia can be looked at you can look at it individually. So then you should look at the hemoglobin in individual cells, which then picks the colors and then make it deep or less. So this is normal chromasia, or even leaning towards hyperchromasia, and this is hypochromasia because there's very little here. So it is the individual hemoglobin here that we are basing on to talk. We are not measuring chromasia. You can't measure it. You have to look at it, So you look at the individual cells to determine whether there's hypochromasia, normal chromasia, or hyper. And that case is dependent on the hemoglobin in the individual cells. And so MCHC, which talks about the hemoglobin in individual cells, is what you should go for. Okay. All right. So that's over now.
I think
there's any more. Yeah.
It's okay.
Okay.
No more questions. Further questions will be received on our platform and then forwarded to you, Prof. So we don't take too much time.
All right. All right.
Thank you.
Fair enough. Okay. So let me see. Actually, we were almost done. Okay, it's a few there. All right. So I'll take myself down and leave the platform for now then. I'll
Okay.
make my way home. Okay.
All right. Thank you.
Have a nice day, Teles.
You
We'll
too.
meet again.
Yeah, bye.
Bye.
All right. So, God willing, tomorrow we are meeting for Campat. Hope to see you all. Bye.
Okay. Tomorrow, Adam. Tomorrow we have a meeting, planning committee meeting around 4.
Okay.
So in case I'm absent, never mind.
All right.
Okay.